Okay, hello everybody. I'm Bill Horsnell from the University of Cape Town, and I'm really lucky today to be hosting Professor Claudia Mauri. Sorry, I've got your name right there, from UCL. Claudia, I'm sure, is really well known to a lot of you for her work on regulatory B cells. She's obviously a luminary in the molecular characterization of these cells, and also backs that work up with really nice complementary human and basic work testing the function of the molecular characterization that she does, particularly in the role of how they will affect autoimmune diseases such as rheumatoid arthritis and lupus, which is obviously very appropriate because she's based at the Bloomsbury Rheumatology Unit. And Claudia's been in London for nearly 20 years now, but she came from somewhere much, well, I think much nicer, from Rome before then. She'd done her PhD in microbiology from the University of Rome, and now she's been in London via the Kennedy Institute and, where I said now, the Bloomsbury Rheumatology Unit. And I think that's enough from me, and I'd like to hand over to the main event to Claudia. Thank you very much, Claudia.
Thank you, and thank you everyone who made the effort to attend today's talk. I took the liberty slightly to change the title just to represent the data that I'm going to share with you today, and is really getting to the bottom of arthritis, trying to understand the link between microbiota, regulatory B cells, and whether we can exploit the microbiota for the cure of rheumatoid arthritis. So let me give you a small introduction about B cells, regulatory B cells in general. So I'm sure that most of you know by now that obviously B cells are extremely important because they produce antibodies. But B cells also are known to produce cytokines. And it was many years ago, about 20 years ago, that Franz Lund come up with this idea to divide B cells according to the cytokines that they produce similarly, in a similar way that we do for T helper cells. And so, for example, what her seminal work shows that if you have a naive B cells, and this naive B cells is put in an environment where there is inflammatory signals, the B cells, naive B cells, differentiate into B effector 1 producing interferon gamma, TNF-alpha, and many other pro-inflammatory cytokines. And these subsets are important for the defense against intracellular pathogens, and they also contribute to the pathogenesis of autoimmune diseases. Equally, if you are in the context, for example, of asthma, where there is, or allergies in general, where there is an abundant presence of IL-4 and IL-13, naive B cells will contribute by differentiating into B effector 2 as well, producing IL-4, IL-13, and so on. My group has now had an interest for probably over 20 years now on the third subset, and I hate defining the subset because B cells are not really like T cells, and they're much more plastic in this way, but B cells can also produce interleukin 10, and when the B cells produce interleukin 10, they are defined as a regulatory B cells, and they play a very important role as immunosuppressant cells. The field has moved on enormously in the last few years, and we do know that regulatory B cells are defined as a subset that primarily, although not exclusively, produce interleukin 10. They can also produce IL-35 and TGF-beta, in particular in the gut. So it's a very much tissue-dependent where and which cytokines they will produce. And via the production of IL-10, they are very good in suppressing the activation of T helper cells, and we and others have shown that they are able to suppress the differentiation of T helper 17, Th1, but most importantly, what a Breg does is to convert, once it gets into a contact with activated T cells, they're able to convey an effective suppressive capacity to the targeted T cells, which then become regulatory T cells. They upregulate the FOXP3 expression as well as the produce IL-10, so they become bona fide TR1. We also show that independently of IL-10, Bregs, via the presentation of lipid antigen as CD1D, they're able to activate invariant NKT cells. And yet again, these invariant NKT cells acquires immunosuppressive capacity. So all of what you see, apologies, I don't have a pointer, but whatever you see on the right-hand side of your screen is very important in the maintenance of immunotolerance as well as the prevention of autoimmunity. But it has become extremely clear in the last few years that regulatory B cells via the production of IL-10 and IL-35, for example, can suppress the activation of cytotoxic T cells, as well as the activation of monocytes, differentiation into macrophages producing TNF-alpha, and they have been identified intratumorally. Therefore, they are, and they've been associated very often in individual cancer patients with a bad prognosis. They are intratumorally, and it's never a good news when they are there because of this incredibly strong capacity to immunosuppress. Now, on your right-hand side, this is a very long old work just to show you that in patients, for example, with SLE, but it's the same with rheumatoid arthritis, MS patients, and so on. Patients with active disease tend to have a very little amount of regulatory B cells, and they don't seem to be, and it seems to be related to the disease status. In other words, when patients are in the inactive status of disease, the regulatory B cells appears to come back, although they never really go back to what we actually see into a normal individual. And just for your knowledge, any healthy individual has roughly 10% of regulatory B cells in circulation. And on the bottom of the screen on the right-hand side, you can see these old slides of ours where we have generated chimeric mice where only the B cells are unable to produce IL-10. And on your right-hand side, you can see that this is in the context of an antigen-induced arthritis. And you can actually see that there is a huge amount of inflammatory cells in the joints in mice lacking IL-10 producing B cells, confirming what is really important concept in the context of regulatory B cells field, that unlike the T cells, we don't have yet discovered an equivalent of natural regulatory B cells. But what we believe at the moment is that they are there to suppress, to constrain excessive inflammation. And they're also present during, in a normal situation during inflammation. But if you remove this break, obviously by removing and producing Bregs, inflammation becomes overwhelming. So one important aspect that we really were interested now for many years was to trying to establish the signals that are important for the differentiation, activation and differentiation of regulatory B cells. And the reason why we are so interested in unraveling these signals is because of what we see in autoimmunity, where regulatory B cells are either absent, like in SLE, there are very, very few of them if you have active disease, or they are functionally impaired, like for example in patients with rheumatoid arthritis, they are numerically as well as functionally impaired. And so there's been a lot of work now throughout the years, and probably one of the most important signals is the activation provided by activated T cells via the CD40 ligands, which activate the CD40 on the B cells, as well as the activation of CD80 and CD86. Work by Simone Filatreau has also shown that the toll-like receptor, for example, is very important if you stimulate B cells via the toll-like receptor 9, and we show via the toll-like receptor 10, B cells then acquire regulatory function and produce IL-10, and they are suppressive. In the last few years, however, it's also become evident, as you can see on the bottom right of the slides, that inflammatory signals, including interferon alpha, IL-1 beta, IL-6, 21, and BAF, are also important for the differentiation of B cells into regulatory B cells. And we postulated and then showed that this is happens at very early stage of B cell maturation, when the B cells comes out of the bone marrow and then transition to stage of development. And if they are encountering the right and correct combination of pro-inflammatory cytokines in addition to activation by T cells, they then will differentiate into regulatory B cells. And on your right-hand side, you can actually see that this is to be very finely tuned, the production of pro-inflammatory cytokines. And in this case, it's just the exposure of this transition on T2 B cells to increase amount of interferon alpha. And you can see here there is an increase of IL-10 as the amount of interferon alpha increases, but that up to a certain level. After that, you see very clear that the amount of the IL-10 decreased dramatically as we increase the amount of interferon alpha to eventually almost zero. Now, so one question that we were thinking about it before this gravel on your right-hand side happened was that what is the role of microbiota in the development of Bregs, or if there is any role at all? And we moved from, as often happens, the answer came from a move from an old building where we had a very efficient animal house, albeit quite dirty, into a new state-of-the-art animal facility where it was so clean that when we moved in, mice didn't develop arthritis. And for anyone who works in this field knows how difficult it is for mice to develop arthritis when you change facility. But most importantly, what we noticed is that these mice, in the absence of developing a disease, they also didn't have any regulatory B cells. And so we postulate, therefore, that there could be a role for inflammation, gut microbiota composition, regulatory B cells, and arthritis. The question that we asked him was whether the gut microbiota was important for inducing inflammations, which then induce the differentiation of regulatory B cells, or if there was a direct function of a role for gut microbiota composition into the differentiation of regulatory B cells directly. And so this is an old slides for a few years ago where we show that in your left-hand side, you see we treat the mice with antibiotics, with a combination of antibiotics known to completely ablate the presence of gut bacteria in mice. And then we treat the mice in the middle with antibiotics. And as you can see here, mice treated with antibiotics did develop a very, very mild disease compared to controlled mice. And what also was important, that these mice had virtually no inflammation as a consequence. And amongst the many different cytokines that we found to be decreased, what the IL-1 beta and IL-6 was the two cytokines that appear to be the most downregulated. And as I said, actually even by giving antibiotics and by eliminating the composition, the gut, the bacteria in the gut, you can see here on the bottom left-hand side that there is a decrease, a profound decrease in the B cells producing IL-10. So there were less regulatory B cells, and purification of the remaining amount of regulatory B cells and transfer into a mouse with arthritis. Mice with arthritis clearly show that they have lost the capacity to suppress compared to those collected from the controlled mice, where they still were able to suppress the development of arthritis in the recipient mice. And so one thinks that we obviously look at whether the regulatory B cells, in addition to be present in the spleens, were also present in the gut. And the answer is yes. They are present in the Peyer's patches, in the ILC, the LPMC, as well as in the mesenteric lymph nodes. But what is very important, and I will stress this concept throughout my talk, is that the regulatory B cells are there at the beginning of arthritis, when there is obviously a bright amount of inflammation. If inflammation becomes too much, then regulatory B cells disappear. And this is in the KBXN mice, which is a model of arthritis, which mimics very closely to what we see is much more chronic, is spontaneous, and therefore is much closer to the human situation. So the right amount of inflammation is needed for the differentiation of regulatory B cells. So the question is therefore, how does microbiota regulate B cell differentiation? Is there direct effect via there is a direct effect, sorry, I need to go back one slide, is the direct effect from the gut microbiota which induce directly inflammation and then leads to regulatory B cells, or can actually propose that there could be also indirect effect and that the microbiota directly or via the production of metabolites induce the differentiation of regulatory B cells. And this second part is really important because therapeutically, what we are facing at the moment is that regulatory B cells are only present when there is inflammation, in a way to protect this overwhelming, to restrain excessive inflammation. But it's very difficult to exploit them therapeutically if you need inflammation and you don't, in a situation like in autoimmunity where you certainly don't want even more inflammation. So having the capacity to activate the B cells via metabolites produced, for example, from the gut microbiota directly into regulatory B cells in the absence of inflammation, it would provide us with a much better tool. And a very small, on your right-hand side up at the corner, you can see that the proof of principle that regulatory B cells do need inflammation, and in particular IL-1 and IL-6. This is an experiment where we generate chimeric mice lacking either IL-1 receptor or IL-6 receptors, and then only in B cells. And then we induce arthritis, antigen-induced arthritis. And you can see very clearly that mice lacking B cells expressing IL-1 receptor or IL-6 receptor develop an exacerbated disease compared to the wild type. The disease was actually incredibly severe. And these mice have also, on your right-hand side, on the letter F, present a less amount of IL-10-producing Breg. So I leave you with this by saying that therefore inflammation, and in particular inflammatory signals, IL-1 and IL-6, are therefore important for the differentiation of regulatory B cells. So we took at that point a step back, and we look at potential molecular bacterial cues that regulate Breg differentiation. And to do that, what we did is to use a model of antigen-induced arthritis and immunizing, took advantage of fate-reported, the vertex mice, and we immunized these mice until they developed disease, and then we took 3 different type of cells, and we perform a microarray. So we purified, as you can see on the top of the screen in the flow cytometry chart, we purify IL-10 positive, EGFP positive B cells, and we compare by microarray the gene expression of this subset compared to the negative counterpart, as well as to the follicular B cells, which we know they never produce IL-10, or they produce much less IL-10, and we took the negative part anyway of this. So we had these 3 populations, and on the left-hand side you can see that they cluster completely separately the GFP positive, what I'm going to refer now from now on as a Bregs, cluster separately from the follicular IL-10 negative as well as the their negative counterpart. And amongst the many genes that they were upregulated, there was one which really attracted our attention, and was the aryl-hydrocarbon receptor. As you can see here, where the red line is, that was upregulated, exclusively present actually in the IL-10 positive and not in the GFP negative, both the follicular as well as the negative counterpart. So why HR is so important, and I will show you a little bit more detail in a second, is because it is a very important transcription factor for that sends cues, environmental cues. But before I move to that, I just want to show you a little bit of confirmation that HR is a transcriptional regulator of IL-10 production in B cells. And you can see here, again, that was done by looking at different transcription factors that have been previously shown to be linked to the production of IL-10. And again, you can see that HR is present in the Bregs, together with the IRF4, has been previously shown by Tomo, as well as BAFT. And many of the others that have been shown to be linked to the production of IL-10 in other cells, including T cells, macrophages, are actually downregulated in B cells. So there is a very specific transcriptional pattern present in these regulatory B cells. And all these signals, in particular HR, was confirmed by PCR. So why, for those of you who are not entirely familiar with the role of HR in immunoregulation, it's known that it's essential for the production of IL-22 in IL-C3. Has been also shown that is important for the induction of IDO in IL-10 transcription in dendritic cells, but also it plays an important role in adaptive immune responses and activate an HR FES, for example, HR activation by FES, which is an HR ligand, promotes Th17 development, but also can induce the differentiation of TR1 by acting in synergy with CMAF to induce the differentiation of IL-10 in B cells. As for B cells, up to a couple of years ago, before we published this data, there was very little known about the role of HR at all in B cells. And there is one paper demonstrating very clearly that, that HR is important for the suppression of IgM response and terminal differentiation of B cells. So we were very lucky that we live across the road of Brigitta Stockinger, who had already generated conditional knockout mice where only B cells lack the expression of HR. And she kindly collaborated with us even before a paper was published. And by taking advantage of the conditional knockout mice, we were able to show that, yet again, the population, the marginal zones and the transitional to B cells, which are the two major populations producing IL-10 during arthritis, you can see here there is a profound reduction of IL-10 in the Bregs population if they lack HR. But most importantly, if we immunize on your right-hand side, immunizing mice with antigen-induced arthritis, you can see that mice lacking HR on B cells, exclusively on B cells, develop an exacerbated disease compared to the control. So the question is, clearly the gut is the main source of HR ligands, and these HR ligands derive from commensal bacteria. And it has been reported that microbiota-derived ligands have been restricted to indole metabolites, but nothing is known, for example, whether other metabolites are involved in the activation of HR. And in particular, I just want to highlight in this review that nothing is really known of the function of HR ligands on the activation of HR in B cells. And so we asked the questions whether microbiota whether this microbiota or microbiota factor play a direct role and bypass the requirement for inflammation in Breg development. And actually, if there was a ligand that activate HR in Bregs and therefore control the balance between pro-arthritogenic and regulatory B cells differentiation. So we took a step back, and what we did is to look at the bacteria that are present during the development of arthritis. And in this case, I'm just showing you what happens at the peak of the disease. And what was very clear is that there is obviously a skew compared to naive mice, but there were a profound reduction of bacteria like the Bacteroideae or the Rickenellas or Lactobacillus that are involved, known to be involved in the production, in the metabolizing of short chain fatty acid. And these bacteria are significantly reduced in mice with arthritis compared to naive mice. Now, the short chain fatty acids are an important source of nutrients for intestinal epithelium cells. And they are known to support the integrity of the gut barrier by providing important nutrient for the epithelium cells. But also, in addition to that, some of the short fatty acids, which the three major ones, acetate, propionate, and butyrate, have been shown to play important role as a cellular mediators and activator of immune cells. And in particular, they have been shown to play a major role in modulating the regulatory T cells versus helper T cells and macrophage differentiation, both at the gut side as well as in the periphery. So the first questions that we asked was whether the butyrate, so sorry, I should say that we treat the mice with butyrate, acetate, and propionate. And we treat both wild type and mu-MT mice lacking B cells, to see whether B cells play any role at all during when we feed mice with these three short chain fatty acid. And you can see here that only butyrate was able to suppress the disease, and this was been previously shown, so that's not new. But what it is new is that if you treat mice lacking B cells with butyrate, we lost the suppressive capacity of the butyrate, and we couldn't find any suppressive capacity of acetate or propionate, and whether the B cells were there or not, it didn't seem to make any difference. Just important to say that we also measure the presence of butyrate in particular, but we also did propionate and acetate, and we found no difference. And you can see here on your left-hand side that butyrate appears to be decreased, significantly decreased in mice with the acute inflammation of disease, but also is significantly decreased in the feces of patients with rheumatoid arthritis. And on your right-hand side, this is what happens in patients with RA. There is a direct correlation between the amount of butyrate that we detect and the numbers of regulatory B cells in circulation. So the more is the butyrate, the amount of butyrate, the higher is the amount of regulatory B cells. Of course, this is an association, so we wanted then to see whether there is actually a direct link between the butyrate and the IL-10. And so the first experiment that we did is to generate chimeric mice lacking interleukin 10. So these mice are unable, the B cells are unable to produce IL-10, and we treat them with butyrate. And as you can see on the left-hand side is when we treat the wild type, there is suppression. The suppression was lost in the absence of regulatory B cells. Therefore, and there was no suppression by acetate or propionate. So that was the first hint and cues that butyrate supplementation require regulatory B cells to suppress disease. Obviously, the second question was, since we have now shown that HR is important for the transcription of IL-10, but most importantly, we know that HR is a sensor for metabolites produced by microbiota, gut bacteria. The next question was whether butyrate supplementation was able to suppress when in mice lacking HR expression on B cells. And the answer is yes. On the left-hand side, you can see that the butyrate is suppressing the MB1 mice, the control mice for this conditional knockout, but again, it's lost the capacity to suppress if the B cells are unable to express HR. And that was also, as a proof of principle, that they do need and require IL-10 ultimately for their suppression, is shown on the right-hand side, where you can see that mice lacking HR B cells treated with butyrate have virtually very, very few B cells, and these B cells are unable to produce IL-10. It is all about regulatory B cells. Well, what it seems very clear is that butyrate supplementation has a profound effect in maintaining regulatory B cells by suppressing the development of a germinal center and plasma blast. So this is a very busy slide, and I apologize that I don't have a pointer, but I'm trying to go slowly. So if you focus on the left-hand side, on the top panel, here we showing the differentiation of plasma cells, and you can see in the first two graphs the controlled mice, and butyrate suppress the differentiation of plasma cells, and on the bottom line there is a profound decrease in the numbers of germinal centers that we found in the actual spleens, and this suppression was lost in mice lacking HR expressing B cells. So to recapitulate this part, clearly butyrate is important on one hand because it promotes the differentiation of regulatory B cells, and it does that by skewing away from the differentiation of B cells towards the plasma, the germinal center, and a plasma cells, plasma blast differentiation. So therefore, the question then that we ask is that, is butyrate directly activating HR, can bind to HR, which then binds to the IL-10 promoters and induce interleukin 10, or it does it in a different way? So there has been a few suggestions in vitro that, for example, HR can bind directly short chain fatty acid can bind directly HR in CACO2 human colon cancer cells. And however, when we do this experiment in vivo, indeed, when we give mice butyrate, we see an increase of CYP1A1, which is the downstream, it gets activated, is a proxy for HR activation. So when HR gets activated, CYP1A gets upregulated, and that's what we see in vivo in B cells. But when we repeated the same experiment by taking B cells in vitro and culturing directly with butyrate, we didn't find any activation of the CYP1A1, indicating us that butyrate is unable to bind directly HR in vitro and in vivo, and so therefore suggests that there is an additional mechanism, different mechanism by which a butyrate activates HR, and that leads to the production of IL-10. So yet again, what we did was to take a step back, and we looked again, and we compared the composition of the microbiota between the naive and between mice treated with butyrate with arthritis and their controls. And first of all, what we notice is that when we treat arthritic mice with butyrate, the composition of the comparative relative abundance of bacteriophile in the feces is normalized, and it looks very similar to what we see in the naive mice. But when we compare the mice, the control and the butyrate ones, what we see is actually the downregulation of so what butyrate does is to increase bacteria that have been previously shown to be important in the pathways in metabolizing tryptophan. And tryptophan is very important because it has already been shown to be involved in the HR activation, but most of the data have been looking at the importance of tryptophan metabolism in the context of carioninin, and what is the effect of carioninin in the activation of, for example, in Th17 and in Tregs. But tryptophan is also important because it has two other additional metabolic pathways. One is that leads to the production of serotonin, and the other one, it leads to the production of indole and indole sulfonic acid. So we therefore query whether also in B cells, one of these three pathways and the molecule involved in these three pathways could be the potential molecules that is activating directly HR. And So what we did is to purify the B cells, and we took the mice and we measured the level of tryptophan. We took the mice, we treat them with and without butyrate, and then measured the level of tryptophan, tryptamine, indole, and carionine, the carionine acid, which is the downstream signals, and the 5-hydroxyindole 3-acetic acid, shortened for 5-HIAA. And you can see by looking at the graphs that we couldn't find any difference. Butyrate did not alter the amount of tryptophan, of tryptamine, neither the amount of indole. There was a modest reduction in carionine, but there was a significant reduction in the carionine acid. But most importantly, what we saw for the first time is that by giving butyrate, we induce an increase, a significant increase of the 5-HIAA, which is the downstream signal of serotonin. So we see an increase of serotonin in mice treated with butyrate. And so to understand better what's going on, what we did in the left-hand side of your screen, we take the B cells and we treated them in vitro with both the serotonin derivative, the 5-HIAA, as well as with carionine A. And you can see that both molecules are able to upregulate CYP1A1, which I remind you is the proxy for HR activation. And this is true in vitro. But what was very important is that only the 5-HIAA was able to upregulate the transcription of IL-10 in B cells. Carionine did not stimulation of the upregulation of carionine that you see in the left, so it binds to HR, but that doesn't lead to the production of IL-10. So suggesting very strongly that the butyrate via 5-HIAA, it is a novel HR ligand that induced the transcription of IL-10. And you can see on the right-hand side that when we treat the mice with either with serotonin or with a carionine acid, only the serotonin was able to suppress the development of arthritis, whereas if it's anything, the carionine actually did, although it wasn't significant, but it was visible enough to induce an exacerbation of the disease. And again, the results in vitro were also supported by the results in vivo. When we took the B cells from the spleens of these mice, we demonstrate that the treatment with serotonin was able to upregulate the CYP1A1 as well as the IL-10, whereas the carionine didn't do so. We repeat the same experiment clearly to prove that this mechanism works via the HR expressed on B cells. We repeat exactly the same experiment where we took the MB1-3 or mice lacking, the conditional mice lacking B cells expressed in HR, and we treated them with the serotonin. And you can see on the left-hand side, the serotonin was able to suppress only and exclusively wild-type mice, but not the mice lacking the B cells expressed in HR, proving that the serotonin inhibition works via require the presence of HR to suppress arthritis. And ultimately, and I apologize, it's a little bit fuzzy, what we did is to prevent the metabolic pathways that lead to the 5-HIAA, and we used two different blocking agents, both the TPH1/2 as well as the PCPA, which blocks in two different stages of development, and I'm only showing the one with the where we block that, and you can see that blocking the development of serotonin will prevent the differentiation of regulatory B cells, and therefore butyrate will not is unable to suppress the disease. So I quite like to conclude with this. I've got two more slides. So one is the cartoon of what we think is happening, and is all work in progress in the lab. So butyrate supplementation increased the amount of HR ligand availability, which by changing the bacteria in the gut. One thing that I didn't say is that butyrate, in addition of the many other published well-known function, is also an antibacterial. So clearly by providing butyrate, feeding mice with butyrate, has increased the amount of tryptophan metabolizing bacteria. And this increased the production of serotonin in disfavor of the production of carnitine acid. And in turn, the 5-HAA activates HR-dependent gene transcription in B cells by enhancing the transcription of IL-10. And this leads to an enhanced Breg suppression, which I didn't have the time to show you today, as well as an inhibition of germinal-center B cells and plasma cells differentiation, which results in amelioration of arthritis. And I'd like to leave you with this historical link between rheumatoid arthritis and gut microbiota, which makes, that's what really made me think even more about the importance of a bacteria composition in the gut, and how the metabolites could be exploited in future for treating rheumatoid arthritis and many other autoimmune diseases and many other diseases in general, by trying to re-establish immunotolerance. And the first one is a very old work in 1909 by a scientist named Carl Worden, who proposed the toxic factors as a cause of rheumatoid arthritis. And he suspected that there was a bacteria in the gut that produced a noxious substance that was absorbed into the body, which then began to attack his own joints. And the way to treat these patients in the older days, apparently, was to give patients to drink the milk, so that you would take the milk, culture, put them in at 37 degrees, and then left it there overnight. And then he would take what we all know, the curd that appears in when the milk goes off, and he would feed patients with that. And apparently that had a very good effect in the disease. And then even more drastically, is on the left-hand side is what is called chronic intestinal stasis. Apparently, Sir Aberton was a very famous gastroenterologist, and he used to remove the whole gut, the whole bowel, when he would encounter patients with rheumatoid arthritis. So indeed, if you can really say, for instance, nothing is more remarkable in the whole range of surgery than the results of removal of large bowel in a case of rheumatoid arthritis. Now, obviously, this is extremely drastic and not advisable anymore, obviously, but it makes, I think, even more, and obviously there is a much more recent study by the Dan Littman group and many others showing that, for example, an increase of Prevotella copri in RA, and an increase of lactobacillus in the fecal, dental, and salivary microbiota of RA. So all of these, I hope that I've inspired you to think more about the function and importance of the gut microbiota, and most importantly of the metabolites, diet, and so on in the future to restore immune tolerance. And lastly, but the most important slides, is the people who have done the work. This work was done in collaboration with Elisabeth Rosser, who began as a PhD in my group and then is now a group leader. And he had all the idea about the microbiota and the serotonin and so on. And the work, the identification of HR as a transcription factors that induce the differentiation of IL-10, regulates the differentiation of IL-10 in B cells, is done by Chris Pipe. I am in depth with Gitta Stockinger, that provided us all the lots of discussion about the role of HR and how the gut microbiota might influence HR activation. My colleagues in Christoph Pock labs in Austria for the analysis of the molecular biologist analysis, and Simon Eaton, who did all the microbiota analysis, the short fatty chain acid analysis, and the funding bodies who have allowed us to perform this work. And I'm happy to take any question. Thank you.
That's fantastic, Claudia. Thank you. Really good. We've got some questions coming through. As moderator, I'm going to ask the first question, if you don't mind. Have you postcoded if there's, I might have missed this in the talk, if there's Bregs actually in the intestine that are directly sensing the butyrate there?
So actually, it's fine. We haven't done it yet. So we, in the paper, we completely focus on the B cells in the spleens. So, which is a good point, because obviously serotonin is produced mainly produced in the gut. I can only say that we think it is, and the fact that we do know that there is a decrease in these metabolites in the gut, and also as an association, there is a decrease of Bregs in the gut with the disease when the disease ensues. So the more, if you have a more inflammation, there is less regulatory B cells. But that is a completely good point, which if it wasn't for COVID, we would already have done that. Yeah.
Yeah. Then the other question I want to ask before I get on to the other ones, right? In the context, has anyone looked at the effect of intestinal? I just don't know, because I don't know much about rheumatoid arthritis. I'll probably find out in a few years. The effect of intestinal infection, so pathological, whether it's bacterial inflammation, parasite, or viral inflammation, on the generation of Bregs, and has anyone looked at how that maybe affects things like rheumatoid arthritis? Okay, so no one has never looked at or found a particular link with a bacterial infection in rheumatoid arthritis, but there is an embargo, so I'm not quite sure whether. But we got a paper in press. I don't know whether the situation, whether you can talk or not, actually. But we have shown very clearly that in both in patients as well as in mice with arthritis, there is an increase of gut permeability, as well as an increase of inflammation in the gut. So, and if you prevent gut permeability by giving it the 1001, which is a compound that restores the level of zonulin on the epithelium cells, so in other words, you reseal the epithelium cells, it might get much better. So obviously, although no one has never found a pathogen present in the actual gut, we do know, for example, that there is that there are bacteria in the joints as well as in the spleen. So obviously, in mice with arthritis, there is a lot of circulation of bacteria or bacterial component which shouldn't be there. They're not there. So you would think it would be due to more bacterial leakage through the epithelium, maybe sort of a peritonitic effect, as opposed to something to do with lymphatic drainage, or maybe it could be a combination of
it is both, actually, yes. So I don't actually think that this is generally speaking for all the autoimmune diseases, I don't actually think it's due to simply an alteration of gut microbiota, but there could be obviously that linked to genetic factors, genetic predisposition, as well as activation of presence of autoreactive T cells and B cells, autoreactive cells, that all together leads to this increase of inflammation in the gut, which is not as overwhelming as you would expect obviously in IBD. So that's probably why patients never really complain much about their guts, but it is sufficient to cause this increase in gut permeability, and then allows the entry of bacteria and bacteria metabolites that shouldn't be there otherwise. that link to the presence of autoreactive cells leads to autoimmunity. I say that's official.
Very interesting to look at. Yeah, we're doing something simple, it's very different context, but yeah, I should look out for your paper. So I want to answer. I just want to find who asked the question because it's an interesting question. I'm going to ask it because I can remember the question. I'm just trying. Yeah. So Maria Mendoza. I'm not sure where she's from. So what's the contribution of Bregs compared to Tregs? If you take the Tregs out, do the Bregs still mediate the effect?
We never done that experiment. We've done it way around. So we took the Bregs out, and the Tregs are profoundly decreased. So in patients with IPEX patients have also have less Bregs. So you would expect that therefore they're very, very closely linked. And I suspect is to do perhaps with the IL-10 and maybe, or maybe because there is too much inflammation. And as I said before, if there is a little inflammation is good, but too much inflammation will suppress the differentiation of regulatory B cells.
Yeah. So you think the Bregs are before the Tregs?
No, I mean that wouldn't go down very well. No, I mean, we never really show. Unfortunately, we don't have an equivalent of FOXP3, and I think Bregs are not like Tregs. It's any B cells can make IL-10. Some B cell subsets make more IL-10, etc. Also, for B cells, you have to think about it as a continuum. It's not like I always view T cells as a very strict, well, not strict, but more or less it's subset. The B cells are in a journey. Inevitably, they will start from immature and they will become plasma cells unless you hit them very, very hard and you block them or you give rituximab to patients and so on. It's just, I always imagine as a journey while they're moving according to the tissue or in the periphery and so on. If there is danger signals that they perceive as a dangerous, it could be, for example, if there is an infection, you see often them an increase of regulatory B cells because you don't want any potential collateral damage. So they're there to mop everything who potentially collateral damage might cause. And so for other situations where you have a little bit more inflammation, but will become plasma cells, and they can, and every stage they make IL-10, including plasma cells.
Okay, and so another question that's been coming through from a few people is, can you see the questions too? What's the effect on the diversity in the microbiota when you lack the Bregs?
Sorry, I was distracted looking at the question. You say hello, people from Ghana. Sorry, say it again.
What's the effect on the diversity of the microbiota?
The effect on Bregs. Yeah. I mean,
or both ways, I think would be good.
Yeah, I mean, so it is interesting because we don't know whether there is a specific, we haven't found a specific phyla yet to species which induce the differentiation of regulatory B cells, but the more diverse, the better it is, generally speaking. And what we found is that an increase, for example, of specific bacteria in certain setting will induce an exacerbation of the disease. And it reduced the amount of regulatory B cells. So we haven't really been looking into the nitty-gritty yet, going deeply into that, because we've got mostly sidetracked by actually, rather than sidetracked, I would say we were more interested on the metabolites. I mean, I'm a microbiologist as a training. I've done my PhD, and I know how difficult, any bacteria can kill other bacteria. They've got millions of different virulence genes, plasmid, and so on. So I always thought from a therapeutic point, it might be easier if we can understand the signals that the bacteria provides with the metabolites and how they influence and alter the immunotolerance, then we can use them in vivo and to target it. And that's what we were trying to show with the serotonin idea.
Okay, so another interesting question here as well from Brian Chung. is there antigen specificity in the Bregs?
Yes, there is. Yeah, so there is. So one very simple experiment that we did many, many years ago was to transfer, we immunize mice with collagen-induced arthritis, and we immunize mice with EAE, with the multiple sclerosis disease. And then we took the mice from collagen-induced arthritis and we transfer into the EAE, and they didn't suppress. Indicating that obviously there is a participation of the autoantigen. And the other experiments that we did is to use the Chris Goodenow mice, where the health mice, and if you have a BCR transgenic animals, they are not Bregs. So they have to be polyclonal first of all, and then there is to be some antigen specificity. However, it remains a conundrum whether the actual antigen is presented to the T cells. We've never done that experiment because we never done it, and no one else has never done it. So there is something that needs to be done.
We tried. Don't know whether it works.
Yeah, exactly. It's the class 2 is important.
Something that's pathogen related.
Yeah.
Whether it was specific, I don't know. Yeah. Yeah. What are the questions we got? Let's have a look. I like David Levy's question. So does the increase in IDO1 metabolism towards KY instead of serotonin have an impact on arthritis?
It doesn't. So we measured IDO1 and there was no difference, and overall chironin had no impact really. As I said, if anything, maybe it was a little bit more an exacerbated disease, but it was not significant.
Okay. What else we got?
What is the status of... Oh, sorry, I took over your moderator.
I'm butting in. Let me be quiet. You carry on. You've got the question there. Go on.
No, I just read this. What is the status of BLIMP1 in arthritis model in HR model? Is inhibited? Is it? I think it's down-regulated. Yeah, it's down on B cells.
Sorry, I just need to plug my laptop in. This seems to be zapping my battery. What else we got?
Oh, there is one interesting from Elisabetta Fabrice about stress, depression, serotonin, and gut inflammation. That is very interesting. Thank you, Elisabetta. So we have started looking in patients with RA taking antidepressants, serotonin reuptake inhibitor. And these are very preliminary results, so it might be not true. But for what we can see, patients who are on antidepressants tends to do much better in terms of clinical score. And there seems to be in remission of rheumatoid arthritis. So we are now expanding this study that was done in a very small cohort, a local cohort. And we're collaborating with our colleagues in Paris with Samuel Biton, who will look at the entire cohort in France. So they have a huge cohort available, and we will do some epidemiological study and see whether is the case, because that would be very interesting.
Yes. Have you so Karina Gomez's, have you looked in detail at the phenotype of the Bregs, and do you get a particular subpopulation or anything come up that's...
We did, and there was any... so no, the answer is we did look at it, but there was no differences. There was not one Breg subtype higher than the other ones. One thing I can say is that HR is expressed in all known regulatory B cell subset, including in plasma, pre-plasma blast, I would say. And any correlation in human? Yes. So we look at it too. There is an HR antibodies that we've managed to just recently to make it work, because otherwise by doing everything by PCR is incredibly painful, in particular in human. And it does work. And I can say that all IL-10 positive B cells are also HR positive, but in human it's slightly more difficult, and there's something else going on and we don't know yet, because obviously we take them from the peripheral blood. There might be that's some of the difference that we see in mouse.
So I think the question by Juan Felipe Lopez is really important.
Do you want to read it?
Yeah. So what's the role of parasites? So I think Porrick Fallon's addressed it. I'm trying to remember.
Oh, yeah.
The work.
Yeah. Atman can. Yeah. So could be the previously described effect. Yeah, absolutely, Juan. That's a very good point. I mean, so first of all, yes, to your first answer, parasites, it was Leishmania in particular, induced, I think, IL-10-producing B cells, and there was a schizosoma, actually, I think. There was a very important, many years ago on MS of patients infected with schizosoma, I think, but please correct me if I'm wrong, showing that these patients had more Bregs and they were in remission from MS. So there has been a long-standing link between parasitic infections and the differentiation of Bregs. And you're absolutely right. I mean, we have no idea what the virome, for example, does. We're all focusing on bacteria because we have to start from somewhere. But I say that, I mean, one thing that we can say is our patients and mice obviously are very clean. So there is not much of parasite, I would think, in the gut of our animals. But and our patients too in London, I don't know. But obviously, another part of the world that could be a very important aspect to study. Yeah.
It's from Jonathan Pollock's a bit of a different question. So any role of platelets associating, or I guess he's talking about maybe platelet association with B cells that.
I don't know the answer to this question. It's incredibly fair, and it's something which we'll definitely should look at it. And in fact, I'm going to write it down because it's, that's a very important question. Yeah.
What else have we got here? Fas ligand.
We didn't look at fast ligand, but I'm getting old and I apologize. I can't remember the name. Sorry. It has been previously shown that fast ligand is present of B cells and in fact Bregs, and these Bregs are called killer Bregs. I'm trying to remember the name. I apologize if you are online. I'm really, really sorry. So we haven't looked at it, whether butyrate increased fast ligand and whether that could be important in their expansion. That is also a very good question. But they do need to be in contact anyway with T cells to suppress. That's very important.
Okay. So, so I think there's a general question from Trishart because we're getting over time now as well.
Yeah.
To make it the last, we make it the last one.
Yeah. So, well, yes, it is. So we know that IBD patients are more susceptible and developing seronegative arthritis. So they don't develop rheumatoid arthritis, but it's often the case that IBD patients present swelling in their joints and they do develop arthritis. So the answer is yes.
Yeah. Cool.
Yeah, definitely shows that. We actually at the moment investigating this in cancer model, because I think that is also very important due to the tumor microenvironment, and I'm expecting that there will be also rich in HR ligand and might have an effect on Bregs expansion in that case.
It's great, isn't it? It's got a really nice discrete role of butyrate here, haven't you?
Yeah.
Lovely. Is that time now? Do we have to finish at 4-ish? Yeah.
I think so. I mean, it was an hour. Yeah, it's very important.
It goes really quickly, doesn't it?
Yeah.
It was great though. Thanks, Claudia, and thanks everybody else. I just need to see if there's any messages from the conveners. There is nothing yet. So that was great, fantastic. I guess people can still keep sending questions through. I've got a message here. No, I haven't. And they'll be fed back to you, Claudia, as well. That's the beauty of this, isn't it? This format.
Yeah, I see there's a lot of interesting questions that we haven't managed to go through, but I promise I will look at them all, and just to understand how I do that, and then I'll try to, in the next few days, answer your question.
Okay, all right. Well, thanks, Claudia. It was great, and hopefully next time it can happen in person.
Yeah.
Yeah. Okay, and thanks everybody for attending. It was brilliant. Thank you so much.
Thank
Cheers.
you.
Bye-bye.